Microbiology Lab Exam Set Number 3. Number of viable yeast cells equals CFUs x dilution factors. What range of numbers of colonies are considered statistically valid? 30 to 300. If the CFUs are greater than 300, there is a probability that overcrowding on the plate could have inhibited some cells from growing. Less than 30 CFUs could involve a sampling error and an underestimate of numbers. Percent viability. Number of viable yeast cells per ml/direct count x 100%. Does a microscope count determine alive or dead cells? Both. Enumeration of bacteria. The yeast Saccharomyces cerevisiae is used in street of bacteria. 1. Count the number of yeast cells in 6 small squares and calculate the average number of cells/square. Count 6 squares that are all next to each other. Each small square accurately represents a volume of 1/4 times 10 to the power of 6 mls of liquid in the counting chamber. To calculate the number of cells per ml, use the equation no cells per ml suspension equals average x, 4 times 10 to the power of 6, 10 to the power of first. The 10 to the power of first is also factored in because the original sample was diluted 10:1. What is the most common method for determining bacterial numbers in a sample? Standard plate or viable count. A sample is diluted in a series of dilution blanks. The dilutions are then plated onto media and the number of colonies are counted after incubation for 24 to 48 hours. It is assumed that the bacterial cells are diluted to an endpoint where a single cell divides, giving rise to a visible colony on a plate. The number of bacteria in the original sample is determined by multiplying the number of colonies by the dilution factor. However, the assumption that a colony represents a single cell is not always correct because cells in a chain, such as Streptococcus, will also give rise to a colony on a plate. Because of the uncertainty in how many actual cells form a colony, count by the SPC are colony-forming units, CFUs. Does the SPC method determine alive or dead cells? Alive or viable? How is the SPC, standard plate count, method biased? Because specific conditions and media are used and these factors may exclude some bacteria in the count. Example, the SPC would severely underestimate the numbers of bacteria in a soil sample because the conditions and the medium used for the count probably favor heterotrophs that grow aerobically at neutral pH values. These conditions do not allow for the growth of anaerobes, chemolithotrophs, or bacteria that may grow at extremes of pH.